It's 11 o'clock here in Minneapolis, so we're going to go ahead and get started. Thank you, everyone, for joining our webinar today. I'm Jackie Carville, the Marketing Director here at ImmunoChemistry Technologies, and I will be moderating this webinar. You may have noticed that your phone has been muted. However, we do encourage you to ask questions along the way. Simply type in your question into the chat dialog at the right and then select Send to Host. We'll answer all these questions at the end of the webinar today. If you have any technical difficulties during the webinar, again, please send your chat to me, and I will do my best to get you up and running. We also will be posting the video recording of this webinar later this afternoon, so look out for an email from me after the webinar with a link to this recording. And finally, we are offering a special discount available only to webinar attendees at the end of the presentation today, so stay tuned through the end for that particular discount code. Now I'd like to go ahead and introduce our speaker, Dr. Christy Strandberg. Christy is a staff scientist here at ICT and was the lead developer of our new Necrosis versus Apoptosis kit. Today she'll be presenting this kit in the context of looking at the overall health of your cell cultures. And with that, I'm going to go ahead and turn it over to Christy.
Thank you, Jackie, for that wonderful introduction. Before I dive into today's topic, I wanted to take a moment to tell everyone a little bit about ImmunoChemistry Technologies. We are a privately owned company that was incorporated back in 1994. We started out by offering custom assay services. These are things like immunoassay design and development, antibody purification and conjugation, and lyophilization. Now, we still offer all of these custom services, but we have since launched several lines of consumable reagents. We have a variety of ELISA solutions. These are things like wash buffers, blocking buffers, assay sample diluents, things of that nature. We also have several different lines of cell-permeant fluorescent probes, and that's the topic I'll be talking about in more detail today. Here at ImmunoChemistry Technologies, we feel that it is incredibly important to understand the overall health status of cell populations that are used in laboratory experiments. We recognize that there is considerable demand for research tools that can allow scientists to determine how their cells are dying. For example, when investigating novel drug treatments, it's critical to evaluate your biological or chemical compound of interest for any unintended cytotoxic effects on your cell cultures. Additionally, oftentimes the goal of a particular treatment is to kill a certain cell type, such as when testing out new cancer therapies. So there's definitely a need for products that are able to accurately assess cell death. Beyond being able to assess cell death as an experimental outcome, it's extremely important to have some reassurance that your indicator cell population is healthy prior to initiating any experimental protocols. Improper cell handling and cell culture maintenance can lead to unhealthy cell cultures, thus potentially introducing artifacts or leading to difficult-to-interpret experimental results. A common mistake is to maintain cell cultures at an inappropriate cell density. Long-term maintenance at too low or too high a cell density can have detrimental effects. But I'll go into this topic in more detail later on in this webinar. The term necrosis has come to have several meanings. Pathologists use the term necrosis to designate the presence of dead tissues or cells. Oftentimes, the term is used to describe accidental cell death that is believed to have occurred in a manner other than through a programmed cell death pathway, such as apoptosis. However, it should be noted that overall, at its most basic level, necrosis is a sum of changes that have occurred in cells after they have died, regardless of the pre-lethal process. Necrosis therefore refers to the morphological stigmata seen after a cell dies and reaches equilibrium with its surroundings. So I want to point out that in the absence of phagocytosis, apoptotic bodies may lose their integrity and proceed to a secondary or apoptotic necrosis. Eukaryotic cells can initiate self-destruction through several distinct programs. These are traditionally broken down into two main categories, non-inflammatory and inflammatory cell death pathways. Both are very important for maintaining homeostasis. For example, for a non-inflammatory homeostasis, think of cell turnover, the normal death that would occur as the cell gets old or worn out or damaged. A normal type of pro-inflammatory pathway would be things like pyroptosis, or a response the cell initiates when it realizes it's become infected or is injured or damaged. Apoptosis involves cellular dismantling that avoids eliciting inflammation. This pathway features activation of initiator and effector caspases. Another non-inflammatory pathway is autophagy. Autophagy features degradation of cellular components within an autophagic vesicle in the intact dying cell. Two examples of pro-inflammatory cell death pathways are oncosis and pyroptosis. Oncosis is an inflammatory pathway that leads to cellular and organelle swelling, which then leads to membrane breakdown, causing the release of inflammatory cellular contents. Pyroptosis also leads to the release of inflammatory contents, but this one is mediated through activation of caspase 1. These 4 different cell death pathways are nicely depicted in a paper published by Fink et al. In Infection and Immunity. I want to take a moment to go into a little more detail on these different pathways. So the first one at the far left, you see apoptosis. As I mentioned, this features activation of initiator caspases. These activate effector caspases, which then go on to cleave cellular substrates. Apoptotic cells demonstrate cytoplasmic and nuclear condensation, DNA damage, formation of apoptotic bodies, exposure of surface molecules, all of this leading to the targeting of these intact cell corpses for phagocytosis. Autophagy also features degradation of cellular components, but in this case it occurs within the intact dying cell in autophagic vesicles or vacuoles. The morphological characteristics of autophagy include vacuolization, degradation of cytoplasmic contents, slight chromatin condensation. The autophagic cells are also taken up by phagocytosis. For the pro-inflammatory pathways, oncosis, if you remember I mentioned before, this is accompanied by cellular and organelle swelling. This swelling leads to a rupture of the cell membrane, which releases its intracellular contents, triggering inflammation. Pyroptosis is unique in that it's mediated by the activation of caspase 1. This is a protease that also activates the inflammatory cytokines IL-1 beta and IL-18. Like oncosis, pyroptosis also features cell lysis and the subsequent release of inflammatory cell contents. So, apoptosis is initiated through 2 different pathways, the intrinsic or extrinsic pathway. The intrinsic pathway is activated following cellular stress, which results in mitochondrial perturbation and the release of cytochrome C. Once released, cytochrome C binds to apoptotic protease activating factor 1, triggering the formation of the apoptosome complex. This results in activation of initiator caspase 9. Once that caspase is activated, it goes on to process and activate the effector caspases 3, 6, and 7, which are responsible for cleaving various cellular substrates. The extrinsic pathway is initiated following activation of death receptors of the TNF superfamily. The trimerized receptor ligand complex recruits procaspase 8 to the death-induced signaling complex via Fas-associated death domain. This leads to rapid activation of caspase 8. Once activated, caspase 8 then goes on to activate the effector caspases 3, 6, and 7, which go on to dismantle the cell. I want to devote a bit more time to giving some background information on caspases, as they are a key component to how ICT's necrosis versus apoptosis detection kit is able to resolve how cells are dying. Caspases are a family of cysteine proteases that serve as critical mediators in apoptotic and inflammatory cell death pathways. Caspases have a primary specificity for aspartic acid residues, cleaving their substrates after a tetrapeptide sequence containing aspartic acid in the P1 position. Caspases are divided into 2 main groups, those that are involved in apoptosis and those that are involved in inflammation. As I mentioned earlier, apoptosis breaks down into initiator and effector caspases. The initiator caspases are caspases 8, 9, and 10, and as the name implies, they're involved in initiating the caspase cascade. Effector caspases are caspases 2, 3, 6, 7, and 13, and these are the ones that are involved in actually dismantling the cell by cleaving various cellular substrates. The inflammatory caspases are caspases 1, 4, 5, and 12. These are involved in the inflammasome as well as cytokine processing. And as we mentioned before, caspase 1 is involved in initiating pyroptosis. Now, to go into a little more detail what exactly pyroptosis is, so during pyroptosis, caspase 1 is cleaved and activated in response to different stimuli, Activation of caspase 1 leads to the rapid formation of membrane pores that allow for dissipation of cellular ionic gradients. This causes swelling and eventually osmotic lysis. The proforms of IL-1 beta and IL-18 are also processed by caspase 1 and are released during pyroptosis. Caspase 1 activity results in cleavage of chromosomal DNA, but I do want to point out that this fragmentation differs from that seen during apoptosis. Nuclear condensation occurs, but the nuclear integrity is maintained. One of the major components of our necrosis versus apoptosis detection kit is a polycaspase FLICA probe. FLICA stands for Fluorescent Labeled Inhibitor of Caspases. These cell-permeant probes are able to traverse the hydrophilic lipid bilayer of cells because of their low molecular weight. They also have an absence of any charged structure, and they're overall rather hydrophobic. All of this leads to them being cell-permeant. All FLICA products contain the same general structure. They contain a unique peptide sequence that's responsible for competing for caspase binding, and that sequence is flanked by a fluorescent reporter tag such as carboxyfluorescene or FAM, as well as a fluoromethyl ketone moiety that's responsible for irreversibly binding to the active caspase. The upper image shown on this slide shows the contents of our necrosis versus apoptosis kit. The lower image shows cells that were duly stained with our polycaspase FLICA probe and hooks. Only the cell in the middle of this image is apoptotic, and that's represented by the presence of the green stain, which indicates caspase activity, as well as many bright blue spots indicating that the cell is beginning to die. The cell in the upper right corner is necrotic. There's no green, and we see scattered blue staining of the DNA. And the cell in the lower left corner is alive but not apoptotic. It's indicated by the lack of green as well as the concentrated blue DNA. The structure shown here depicts this polycaspase targeting peptide sequence. This is our FAM-FLICA structure. The polycaspase targeting sequence is valine, alanine, and aspartate, or VAD. These amino acids are flanked by carboxyfluorescene as well as the fluoromethyl ketone moiety I mentioned previously. FAM-FLICA, this polycaspase amino acid sequence, has an affinity for most caspases. This includes caspases 1, 2, 3, 6, 8, 9, 10, and 13. Here's a high-level overview of how we believe the FLICA probe works. The reaction I'm going to talk about is what we believe to be the most likely mechanism of caspase inhibition. We traditionally think of this as two reactions, a slow reaction followed by a fast reaction. Once the inhibitor sequence, that valine, alanine, aspartic acid sequence, is recognized by the caspase's reactive binding site, this is what we consider to be the slow reaction, the time it takes for that sequence to find the active caspase. Once the slow reaction has occurred, then we have an almost immediate fast reaction where a covalent bond is formed between the FLICA probe and the caspase. So what I'm showing you on this slide here is a reaction where the active site thiol present in the catalytic site of the active caspase reacts with the carbonyl carbon of the FMK moiety on the FLICA probe. This results in the formation of a thiohemiketal intermediate. When the active site thiol reacts with the carbonyl carbon, a covalent bond is formed via a three-membered ring. This three-membered ring rearranges to give rise to a final thioether adduct. The end result here is an irreversible bond between the caspase enzyme and the FLICA probe. So overall, kind of in the bigger picture, what this means is that when the caspase is in its inactive zymogen form, the catalytic sites are blocked. Once this is processed to its active form, these catalytic sites become exposed. The FMK group on the FLICA probe is then able to form a covalent bond with the reactive catalytic site on the active caspase enzyme. This enables FLICA probe to be retained within the cell despite any subsequent wash steps because they're no longer membrane permeable once they're bound to the caspase. It also means that FLICA won't detect inactive caspases. It has to be in its processed active form to react with the FMK moiety. It's also important to note that FLICA is not cleaved by the caspase. Once the FMK reactive groups on the FLICA probe form a covalent bond with the two catalytic cysteine groups within each of the two caspase reactive sites, that particular caspase enzyme is essentially rendered inactive. It can no longer cleave any substrates. Here are some sample flow cytometry results showing how FAM-FLICA alone can be used to detect the number of apoptotic and non-apoptotic cells in any experimental treatment. The graphs on the top are showing the forward and side scatter results as well as the fluorescence intensity of Jurkat cells that were mock treated with DMSO. The lower graphs show the results from Jurkat cells that were induced to become apoptotic by exposure to 1 micromolar staurosporine for 4 hours. You can see a noticeable increase in the FO1 signal when you look at the induced cells on the bottom compared to the mock-treated cells on the top. When cells were exposed to just DMSO, the majority of them, 92.1%, did not stain positive for active caspases. In contrast, when the cells were exposed to staurosporine for 4 hours, we see approximately 96% showing signs of having active caspases. I want to switch gears briefly and introduce the other main component of the necrosis versus apoptosis detection kit. 7-AAD is a membrane-impermeant far-red dye that intercalates in double-stranded DNA. 7-AAD has a high affinity for cytosine and guanosine-rich regions. Normal healthy cells that have intact cell membranes are able to exclude this impermeant dye. So 7-AAD stains the nucleic acids red in cells that have compromised membrane integrity. The optimal excitation for 7-AAD is between 485 and 495 nanometers. The optimal emission is between 515 and 525 nanometers, making it compatible with our green fluorescent FLICA probe. Here are some sample flow cytometry results showing 7-AAD staining of live untreated cells compared with cells that were killed by a brief exposure to 90% ethanol. Overall, the live cells that you see on the far left graph, they're able to exclude the 7-AAD dye because their cell membranes are intact. Meanwhile, the cells that were killed by exposure to ethanol, the majority of those are staining red, and they've taken out the 7-AAD dye. Their membranes are no longer able to exclude that dye. If you mix these two cell cultures, the live cell sample with the ethanol-killed sample, as you can see in the third panel at the far right, you can detect both populations, both the dead and the live cells simultaneously. This combination of ICT's FAM-FLICA with 7-AAD allows for an enhanced understanding of cell death. This combination allows for the simultaneous detection of 4 cell populations. Our necrosis versus apoptosis kit is able to detect the live healthy cells, cells that have entered into early stages of apoptosis, cells that have entered into late stages of apoptosis, as well as cells that have died. Any live non-apoptotic cells will remain unstained as they do not contain active caspases, and they also have intact cell membranes which are capable of excluding 7-AAD. Cells that have entered into early apoptosis will fluoresce green with FAM-FLICA as they contain caspases that are active, but they will not fluoresce red as 7-AAD is excluded by the intact cell membrane. Cells that have entered into late stages of apoptosis will fluoresce both green and red due to the presence of active caspases as well as the loss of cell membrane integrity. Dead cells will fluoresce red only as they do not contain active caspases and they've lost their membrane integrity. If you're analyzing by flow cytometry, when you plot the FL1 channel by the FL3 channel, you can easily identify these 4 cell populations as they show up in separate quadrants. The lower left quadrant will house the live unstained cells. The lower right quadrant will contain the green staining early apoptotic cells. The upper right section will contain the green and red late apoptotic cells, and the upper left section will house the dead cells that have stained red only. I'm showing you a couple sample graphs of either untreated Jurkat cells or mock treated with DMSO, or cells that were treated with 1 micromolar staurosporin for 4 hours to induce apoptosis. When we compare the mock treated and the staurosporin treated cells that have been duly stained with FAM-FLICA and 7-AAD, what we see is that the majority of the mock treated cells fall into that live unstained category. Well, very few of the induced cells, star sporin-treated cells, remain in this category. Roughly 46.3% of the star sporin-treated cells have entered into early apoptosis, and approximately 50.6% have entered into late apoptosis. As I mentioned earlier, improper culturing techniques can lead to unhealthy cell populations. So I wanted to touch on that briefly. This necrosis versus apoptosis detection kit can easily be used in labs to periodically check to make sure your cell cultures are healthy. Here I've intentionally allowed Jurkat cells to become overgrown prior to staining them with the necrosis versus apoptosis kit. Cells that were maintained at concentrations ranging from 1 × 10⁵ to 6 × 10⁵ cells per mil appear to be healthy, as most of the cells are located in that lower left corner, which is the unstained live healthy quadrant. As the cell cultures were allowed to reach concentrations of 1 × 10⁶ cells per mil or greater, we see a pronounced increase in the number of cells entering early apoptosis, as evidenced by the population shift that's showing an increase in FL1 signal. As you can see, if you compare the 1 × 10⁶ graph to the ones with lower cell concentrations, we see a drop. We're dropping in the percent of the live healthy cells. It drops from roughly 80% down to just below 60%, and we see an uptick in the early apoptotic cells. And that's even more pronounced as cells reach 3 × 10⁶ cells per mil density. This trend is also evident for adherent cells. Cells that were not allowed to surpass 80% confluence are shown in the graph on the left. Very few of these cells fall outside of that live, healthy lower left quadrant. Cells that were allowed to grow and reach near 100% confluence show an increase in the percent of cells entering both early and late apoptosis. In the cells that were cultured at a less than 80% confluence, about 91.5% of the cells fall in that live, healthy quadrant, and in the cells that were allowed to become overgrown and are near 100% confluence, only about 74% of them are healthy. We have just about 15% entering early apoptosis and close to 9% entering late apoptosis. The necrosis versus apoptosis kit is also compatible with fluorescence microscopy analysis. This slide shows images captured of Jurkat cells that were treated, again, with star sporin for 4 hours to induce apoptosis. Here you can see early apoptotic cells that are staining green only, cells that are in late apoptosis as they stain green and red, and cells that have died as they stain red only. So whether you're interested in assessing the overall health of a particular cell population, monitoring cell death or cytotoxicity as an experiment endpoint, or simply want to obtain a more detailed understanding of the number of cells that are healthy, dead, or in different stages of apoptosis, we encourage you to try our new necrosis versus apoptosis detection kit. I also want to take a moment to remind our listeners that ICT offers a variety of fluorescent probes. If you have any questions about other products or services offered by ICT, please contact us. I'll post our contact information on the next page. We offer a variety of fluorescent probes for detecting active Caspases. We also offer kits for detecting cytotoxicity, as well as products that are capable of monitoring Caspase activity in real time. We have a panel of products for detecting oxidative stress, as well as monitoring mitochondrial membrane potential. We can also provide cathepsin detection products, serine protease detection products, and various nuclear stains. If you have any questions or want to browse our product list further, I encourage you to call us at 1-800-829-3194 or visit our website, www.immunochemistry.com. Or if you have a particular question, you can either type it into this webinar or contact us through help@immunochemistry.com. And with that, as Jackie had mentioned, we are offering a special promotion for any of our webinar attendees. You guys can take 25% off your order of the Necrosis versus Apoptosis detection kit. This is valid from now through the end of February. So with that, I would like to open the floor for any questions.
Wonderful. Thank you so much, Christy. We do have a few questions coming in here. Again, you guys feel free to chat those questions into me. Under the little chat dialog, select Send to Host, and we can either pass those on to Christy or, depending on time, follow up with you after the webinar. So the first question that we have here, someone's asking about the sensitivity of FAM-FLICA. So how sensitive is this?
That's a great question. Our FAM-FLICA product is very sensitive. This reagent can detect natural apoptosis occurring in control cells. If you look back to some of the slides I've shown where we have the mock-treated cells, we do see some of that population. Typically, between 2 to 6% of the population will naturally enter into apoptosis. Our FAM-FLICA product is capable of detecting even that naturally occurring apoptosis.
Wonderful. Thank you, Christy. The next question, somebody with reference to this particular assay kit, they're wondering how long does the assay take?
Absolutely. Another great question. So this is a type of experiment that can take place easily within the course of the day. Staining with FAM-FLICA, and we encourage that you stain with FAM-FLICA first and then stain with 7-AAD. The FAM-FLICA reagent starts to react within 15 minutes of addition to the cells. Typical time window for staining with FAM-FLICA falls between 15 minutes to 60 minutes. Although we have had end users incubate their cells with FAM-FLICA for up to 72 hours and didn't see any detrimental effects on their cell cultures. The staining window for 7-AAD is between 10 to 30 minutes. The exact incubation time for both reagents needs to be determined by your experimental protocols. It could be different for different cell lines.
All right, thank you. So someone else is wondering what types of instruments are compatible with this particular kit?
We recommend fluorescence microscopes or flow cytometers for this kit. Certainly it could be adapted to other applications, but at this point we've just tried it with those two instrument types and provide protocols in our manual for those two types of instruments.
Great. Somebody else is wondering, can their cells be incubated with the Necrosis versus Apoptosis kit for an extended period of time?
Sure, good question. As I mentioned earlier, we have seen that people can incubate their culture cells in the presence of FAM-FLICA for up to 72 hours without seeing any detrimental effects. I would not recommend a prolonged exposure to 7-AAD as this compound is toxic to cells over time. If you know that your experiment is going to be a longer time course, I recommend designing your experiments with the idea that you'll add 7-AAD at the very end, just prior to analysis.
Great. We also have another question here about fixed samples. So is this kit compatible with samples that have been fixed?
Absolutely. As long as you can stain your cells with FAM-FLICA and 7-AAD prior to fixing them, then this kit would be compatible with fixed cells. Since FAM-FLICA only binds to active Caspases, this kit would not work with cells that were fixed prior to staining, as this process of fixing the cells would inactivate any Caspases that were present. ICT does include a formaldehyde solution in the Necrosis versus Apoptosis kit. This solution is designed to cross-link and aggregate cellular components and will not interfere with the carboxyfluorescene or the 7-AAD labels. We recommend that any end user avoid using absolute ethanol or methanol-based fixatives, as they will inactivate that carboxyfluorescene signal.
Great. And then I think, just for sake of time, to keep this under half an hour for everyone's busy day, we'll do one more question here, and then anyone else you guys can keep chatting in the questions, and we will follow up with you right after the webinar. But I guess to take a last question here, someone wants to know how many tests are included with this particular kit and what constitutes one test?
Sure. So we estimate that each sample or each test will contain a volume of 0.3 mils or 300 microliters. We offer two sizes of the kit. The trial size contains enough FAM-FLICA and 7-AAD reagent to assay between 50 to 100 tests by flow cytometry, or roughly 50 tests by fluorescence microscopy. Oftentimes, you can get by with a lower staining concentration for flow cytometry, so that's why you can extend the number of tests to a higher number with flow cytometry, and probably not as easily done with fluorescent microscopy. If you're staining larger cell volumes, the trial size kit provides enough of these reagents to stain between 15 to 30 mils of sample. The standard size kit contains enough FAM-FLICA and 7A-AD reagent to assay between 100 to 200 tests by flow cytometry, or roughly 100 tests by fluorescence microscopy. If staining larger cell volumes, the standard size kit provides enough of these reagents to stain between 30 and 60 mils of sample.
Great. Thank you so much, Christy. All right. Well, that's about all the time we have for today. So again, if we were unable to get to your question, or some of the questions I'm seeing require a more in-depth technical response, we're going to follow up with you personally right after the webinar. And again, I will be sending out the full recording of this webinar within the next day. We're looking at our next upcoming webinar sometime in early March. That will be on some ELISA solutions. So check back soon on our webinars page, and we should have that up and available for registration. If you can think of any other questions after the webinar or have ideas for future webinars, please fill out the Contact Us form on our website or reach out to us through social media on LinkedIn, Facebook, or Twitter. And thank you again, everyone, so much for joining us for this webinar. Have a wonderful day.